All right. Hello, everyone. Welcome to today's SONA webinar series. I'm really excited to be, to have Thomas with us today. Thomas is a colleague, and he's an exceptional, uh, promising scientist from Ghana, which is close to Nigeria, where I come from. And he'll be talking to us today about blood phosphorylated tau as biomarker for Alzheimer's disease. But before going into that, let me give you a brief bio about Thomas. So Thomas is an assistant professor for neurochemistry at the University of Gothenburg in Sweden. He holds a research fellowship from the Bright Focus Foundation and is the principal investigator of grants from the Swedish Alzheimer's Foundation, the Swedish Brain Foundation, the Swedish Dementia Foundation, and several other bodies. His main research interest is to understand the processing of tau protein in Alzheimer's brain and apply this knowledge to develop new CSF and blood biomarkers for clinical use. To this end, he has approximately 40 publications and a pending patent. And Thomas has received two major awards for his discovery and development of blood p-tau biomarkers for clinical use. First, he was co-awarded the Best Scientific Discovery Award in Alzheimer's field in Sweden for over 2019-2012. And secondly, he won the Astrup Prize for Contemporary Work in Clinical Chemistry, awarded by the Nordic Federation for Clinical Chemistry. Thomas was awarded his PhD in 2018 from Warwick University in Coventry, UK, funded by the BBRC. At the end of his PhD, he was a visiting scholar at Harvard Medical School with fellowships from the Royal Society of Chemistry and BBRC. And previously, he had completed his bachelor's education in biochemistry in his native Ghana with first-class honors and was awarded a Dean's Award for Academic Excellence. He followed that with Masters in Pharmaceutical Biotechnology in Leicester, funded by Commonwealth. It's really exciting to have Thomas because he's someone that is a rising star from Africa. And whenever I see him or I talk to him or I see his papers, it reassures me that someone like this will be doing these great things. So it's really exciting to have him today with us. Thomas, go ahead and give us your presentation. And for the viewers, please, if you can drop your questions in the Q&A section. And for those that are on Facebook, you can drop your questions as comments. I'll try as much as possible to go back to Facebook and get them. Thank you.
Thank you very much, Mahmoud, and thank you for having me. And also thank you for organizing such a nice series of webinars. I've been following some of them, and it's very good to be getting in touch. So I'll be talking today about phosphorylated forms of a protein called tau in the blood and the possibility of using these as biomarkers for Alzheimer's disease. The organizers asked me to give a little bit of background about myself, so I just thought I would start with that. But Mahmoud has basically said everything. This be my career trajectory. From 2010 was finishing my undergrad in Ghana, and then 2012 masters, and then I went back home to Ghana and started a position as a lecturer for a while in a polytechnic, and then came back to complete my PhD. I had my viva end of 2017, but I was awarded in 2018, and then started a postdoc at Harvard and at Gothenburg, and I've stayed here for the last 2 years and now transitioning into new positions. So hopefully there might be some more coming out later. So when it comes to Alzheimer's disease, it's a really common name that we know when it comes to dementia research, but the whole basis has been with a doctor called Alo Alzheimer ages ago when he observed this patient called Augustus Dittem in Frankfurt in Germany. So that's the first patient of Alzheimer's on record. Now, since then, the Alzheimer identification has been based on clinical syndrome, so based on clinical symptoms and signs, and usually is either the individual has preclinical, like asymptomatic. So they have maybe biological evidence of the disease, but clinically, they don't show any cognitive signs that they're declining in their cognitive abilities. But that is quite difficult to identify in the clinic, and oftentimes it might be missed. For individuals who just started from the clinical stage, so that's the mild, so they have mild cognitive impairment, and then later they go into full-blown dementia stage. Now, due to these underlying changes that go on in the brain and you reflect in some other parts of the body, there's been a focus on biological changes because these ones are more difficult to miss. It's either there or not. So there's been a two-way interaction between clinical and biological. And now we're looking at using the biological evidence to support the clinical and vice versa. Now, in the biological side, it's mostly two main pathological phenotypes that are used as evidence of Alzheimer's. That it's either plaques in the brain, so these are sections through the brain, so the plaques that contain amyloid beta protein, and tangles that contain phosphorylated tau, which is main what I'm going to talk about today. And that's identified by neuropathology. So where at the end of, like, when someone dies, as postmortem, then slices of brain are taken in. And because that's only done at the end of life, during life, what are ways that we could try to identify the disease? There are some ways, including brain imaging, and there are also CSF testing, CSF biomarkers. And now we're looking at, could we try and look at blood biomarkers as well? So this is the trajectory of Alzheimer's disease. As I started talking about, we have individuals who are normal, cognition, nothing changed in them. And then people who have mild cognitive impairment, and then they progress to the dementia stage. But before all these things come up, there are biological changes that are occurring that are not easy to identify. And these ones are in the form of amyloid beta. So you have either CSF or measured by brain imaging, and that you get to have tau images changes occurring later, which is something that gradually we feel that what we think about the time difference between amyloid and tau might need to be changing. And that there also degeneration of neurons. So neurons start to degenerate, that the ability to send information between themselves get to be affected. And these biological changes do underlie the changes that then lead to the cognitive decline that we see in patients who become forgetful. They have some changes in the executive function and all that. Yes, and that's when the dementia sets in. So because of these biological changes that we know, there's been a criteria for the diagnosis of Alzheimer's that has been proposed by the NIH together with the Alzheimer's Association in the US, is that you look for three things, and they call ATN criteria. The A is the amyloid, as I talked about. Either you measure that with brain imaging, which is PET imaging, so you give a radioligand to an individual, and that ligand crosses the blood-brain barrier, it goes into the brain, and then you do a scan, and if the individual has aggregates of amyloid beta, then it will light up. If they do not, then that, you get to have a much lower binding. Or you use CSF, where there's an inverse relationship between the amyloid beta in the brain and what comes into the CSF. So it reduces with increasing disease. Or you use tau. Same, you do imaging with a PET radio tracer, or you use CSF. Now, the main one that is being used currently is phosphorylated 181 in CSF. But here, the amount of it increases rather with disease. So from control to MCI to disease to dementia, you get to have higher levels. Similarly, we have neurodegeneration, and it's either you do MRI, that in MRI, you get to see atrophy of the brain in someone who has Alzheimer's disease. And then if you use total tau or neurofilament light, you get to see increasing levels of degeneration of neurons. So because of these three core criteria, that's what I'm going to talk about as to how we can use biological basis to identify, to diagnose and prognose Alzheimer's disease. So if we have these markers, we have imaging markers, and we have blood CSF markers, why do we then need blood? Well, for several reasons. When it comes to PET imaging, just the positron emission tomography, which uses, the sort of CT scan, MRI scan based approach, that you give a ligand and then it goes into the brain, is really expensive. That in several settings, even in developed countries, you cannot access them everywhere. That is limited to a few specialized centers. So it's quite difficult to get access onto these ones. And oftentimes, because they focus on aggregates, and aggregates form later in the disease spectrum, they tend to be more useful when the disease is a bit more established. So you might miss the preclinical phase. And some of the ligands do have off-target binding. So you might see false positives. And CSF is a bit better in terms of approach, that is cheaper, but then you still require lumbar puncture, and you require someone with the training to do that, and it's quite invasive. So the one one of the major problems is that they have like poor accessibility, especially in settings where you don't have, the luxury of conducting these markers. So this is not the first time we're talking about blood. People have looked at it for a long time, but the problem is that it is complex to look at blood because, of course, you're looking at the brain, which is the primary tissue of where everything is occurring. But from the brain, you go down to CSF, and then in CSF with the cerebrospinal fluid, because that's in really in close contact with the brain extracellular matrix, and then you can go to blood. You lose proximity to the disease. So the farther you move away from the disease source, you also loosen concentration of the analytes that you want to measure. However, we're looking at access, as I just said on the previous graph, we have a higher access of blood that's more accessible. But also there's also proteolytic processing because your target gets to be processed multiple times. So you're likely to have difficulty measuring what you want. So there've been initial assays like amyloid. As I said, we have amyloid, we have neurodegeneration, we have tau. So A, amyloid, T, tau, and neurodegeneration. There are amyloid markers in blood that have been identified. Their performance are not so well. And there are also neurodegeneration markers, including NFL, that was also developed by my lab, that it's really good. But one problem is that NFL is not specific to Alzheimer's. You see it increasing in any form of disease, neuronal disease that has degeneration of neurons. So whilst it's useful, it doesn't tell you someone has Alzheimer's. And so this moves on to phosphorylated, which we know is really specific. There've been new assays earlier that people developed. This was quite a nice one, for example, one that was published in 2017. But the problem was this: they was not sensitive enough. There were a lot of individuals that were given zero, practically sitting on the zero, which means you cannot really tell if these individuals truly have, because zero means that you cannot measure. So we want more sensitive assays. There have been other ones. This was published earlier. It's been improved and was published recently in a much better form. There's another one that was developed with a different technology. But whilst they show an increase in Alzheimer's, they also show increases in practically all the other dementias that you see. However, from CSF, we know that if you're measuring phosphorylated 1A1 at least, it is specific to AD. So if these are not specific, then there must be something happening that we understand. So this is where I come in. We know that the CSF markers, so the current CSF markers that measure phosphorylated 1A1 and total tau, these are amino acids that were built in this very lab by my mentors in 1993. That what they show is that they use amino acids. This is a tau, so this is a cartoon of the tau protein, and that they use antibodies that target the middle of the protein to measure the phospho, and same also to measure the total tau. But if you use these ones, you don't get in blood, and that's been where we've been ending until recently. But what from an experiment that was not working for me, what I got to find out was that if you start from brain, you have a lot of proteolytic processing, so some chopped off parts. However, if you piece them together, you get a whole sequence of tau. But once you move away from the source, which is move away from the brain, if you come to CSF, then you start losing some shaving is occurring. You're losing some of the C-terminus part. And that once you move away further to blood, you lose even more in terms of the coverage and also in terms of concentration. And this appears that it means that if you're looking at assays to measure in blood, you should not be looking at the mid region here that we know in CSF. You should be looking at the other side here, these sides, the more end terminals, the farther end going back to the left. And that is the key that we just got, and that's been the basis of it. Now, also we used an amino acid platform, unlike ELISA or Western blots that are not very sensitive. We use an amino acid platform that's highly sensitive. This is a bead-based platform. You have beads, and then you coat your capture antibody onto it. They form an amino complex, and you add your antigen, so your blood that you want to measure, and then your antigen from the blood will bind onto this. And that once you have that immunocomplex, you get to wash several times, and you add your detection antibody, and then they get to be so these beads go into wells. So those are the kind of wells. So each well is only big enough to carry one single bead. So it goes in there, and then after everything, they're sealed and they're measured. They're imaged actually. So there's an image, and these ones are the wells. And the lit up parts are those that are positive, showing positivity for your marker. And based after the imaging, you go through some statistical calculation, and then it gets to give you a value, and you measure in this instrument for. So usually after you have everything together, it's quite semi-automated. You slot it into this instrument, and then it works out for you what you want. There are commercial assays that you could buy to not plug and play, but we also develop some from scratch, which is, for example, in the phosphatile assays that we have developed from scratch. So one of the first things we try to understand is that in blood, which forms of blood are useful. That we first show that in plasma, if you use plasma or serum, they are both useful. So here we use the same individual, so paired individuals. The same individual took EDTA plasma into serum, and then you show that in those older adults, like A beta negative older adults, who were negative, like controls, they had no signs of dementia. And if you compare those to those with Alzheimer's disease, there were an increase, and that the accuracy, so there's like an AUC curve, area under the curve plots, that it tells you that they're about the same, and then there's quite a good correlation between them. And more recently, we've looked at if you use different types of blood metrics, that if you use EDTA plasma, if you use serum, if you use heparin, if you use citrates, they are measurable. However, they affect how much you're measuring. So if you use EDTA, which is often used as the basis, and you compare all the others to it, you see like you have a drop in serum, you have an increase in heparin, and you also have a drop in citrate plasma. So it means that the source that you use, the type of blood that you use, has an effect. But the good thing is that our methods are able to measure regardless of which type of blood you use, which is very good. So we looked at the diagnostic performance, that in one of the biggest cohorts, this is from McGill University in Canada, that we show that it starts, so in young individuals, young adults about 20 years of age, we're able to measure really tiny levels of blood levels of this marker. It increases a little bit, not significantly, in older adults, but then it doesn't stay there. Once you have amyloid positivity, so amyloid changes that happen if you're positive for amyloid beta, the levels go up significantly. And that, once you transition to cognitive decline, you get to see positivity in those who are positive for amyloid beta in cognitive declined individuals. But if you don't have it, you still stay at the same level as you have for controls. If you have Alzheimer's dementia, then you go way higher. So it just validates what we think we're looking at. And then we show that it correlates with the levels that we have, correlates with CSF levels of tau PET, so that the aggregates of tau, and that we measure in the brain, you get to see quite a good correlation, just to show that this includes both negatives like amyloid, those who have no amyloid pathology, and those who do have. So it pushes it a little bit. But if you focus just on those with a beta positivity, it gets way better. And that when you compare with PET, you can see that they correlate with increased levels of tau aggregates in the areas of the brain that are known for tau tangles. And similarly, we show same for amyloid beta. And that it appears that from blood, you could predict what's going to happen if you use PET. So individuals who were negative for tau PET compared to those who were positive for tau PET, if you measure blood phosphatile in these, you had higher levels in those who were positive for tau PET. So it means that it appears that levels do happen. And even if you do the same for amyloid, you show that those who have higher levels of phosphatile have higher levels of amyloid positivity. And that it could change ahead of PET. That this Bragg staging is a way that individuals who have gone to postmortem, this is how pathologists do stage how the extent, the intensity of the pathology of tau that they see. That if you're stage 0, it's from 0 to 6. So stage 0 here, we have a lot of it. But then as you go along the spectrum, you have increased levels of blood phosphatile, which is quite a good thing. And even within this group, so the group that has no tau positivity, so they have no tau aggregate in the brain. But if you look at those ones and then you split them, you can see that there's some who have increased levels of blood phosphatile. So potentially this happens ahead of PET. And that they can predict future cognitive decline. So here is a group that we looked at changes before and after. So at 1 year, and then a year later, these patients were assessed again, that you can see that those who had higher levels of phosphatile also had higher levels or higher levels of cognitive decline 1 year later, which is 1 year is a really short time to see cognitive changes. But this is a little bit messy, but it's quite nice that I would like to show. These are individuals who were started being monitored for changes from the preclinical stage. So no biological evidence of AD, clinical evidence of AD. And so all these graphs are cognitive changes, so different tests that have been performed on them to look at their memory or another cognitive domains. That when you look at it based on blood measures, it's only individuals who had increased levels of A beta positivity and phosphatide positivity. Those are in the red. So in all the plots, you see the red going down. So these individuals decline on a mean level, on average level, decline with their cognition over 5 years. So there's a point where they were taken, the blood was taken. So they are assessed for 7 years, but 5 years after blood results, you can see that those were declining a lot more. So it gives you an idea that you can use this to predict what's going to happen with someone years later. And similar to CSF, we show that the markers are specific to AD. That, for example, if you compare to vascular dementia, which is quite a common comorbidity found in Alzheimer's, you see that the marker does not, it's separate those with AD and those who have only vascular dementia. And these are others like Parkinson's disease, frontotemporal dementia, PSP, progressive supranuclear palsy, or other forms of tauopathy. So these have tau. So the same ways of identifying tau at pathology for Alzheimer's, the same way that we do for these ones, except that they usually don't have amyloid. And you said that the marker can tell you who specifically has Alzheimer's and who might not have Alzheimer's. And we've shown the same thing in other cohorts. And then when you compare to other markers, so these are like AUC plots. I'm going to show a lot of this, so I suggest people like get used to it. So these are plots that gives you an idea of the accuracy for a marker to separate between groups. So between Alzheimer's disease dementia in individuals and those with cognitive cognition intact, that you can see these are like fractions. So you can see it's like 91%. So that you can see like compared to a lot of other markers in blood that have been used, you have way higher levels. There's higher confidence in phosphatide compared to the others. But all these ones we've been talking about are in sporadic AD. So individuals who have AD that we don't know the cause. It could be due to multiple sources of causes. What about those who have genetic predispositions to Alzheimer's at a later age? And this is one group that we did. This is based on a collaboration with UCL, where so non-carriers, the controls who have no disease, but those who have genetic mutations that predispose them to the disease, but they are not showing symptoms yet, they already have a higher level. And those who have genetic predisposition but have a higher level and showing clinical symptoms already even has much higher level. And that if you do a model, you can show that is about 16 years prior to changes. So right from here to there, 16 years up to 16 years, you can confidently say someone is going to have a predisposition to Alzheimer's, or they're going to develop Alzheimer's in the future. And then we looked at Down syndrome. Down syndrome is quite a debilitating disorder, which has also a genetic predisposition to Alzheimer's. Similarly, we show that those who are asymptomatic, those who are pre-symptomatic, and those who have the dementia stage, you have an increase in level going there, and that you can show quite a good AUC, so accuracy to separate them. And so we've talked about ways in living individuals, but then if you want to show a marker is really good, you should be able to say that it can predict postmortem, because at present, postmortem pathological identification is the only sure way to say someone truly had AD. Well, unfortunately, that's too late to do anything for any interventions. So remember that first I showed this slide, showing like you have plaques and you have tangles. But these come from, you take brains from individuals, they take slices, and you already see like there's a lot of atrophy. So like waning away of brains brain areas. And then you get to see these are the tangles, and you get these are the plaques already there. So what we did was that we took quite a unique cohort from King's College in London, where individuals had been followed for several years. We were fortunate to be able to get those who had been followed for up to 8 years and blood taken around that time. So we could say that if the person, the diagnoses that were given to them during their life, and then compared to that the blood markers that we analysis that we do and compare to pathological diagnosis that was given after death, what is the surest way to diagnose the disease. So this first what we saw. When we plotted, this is the diagnosis 8 years before, 4 years before, and 2 years before death. And this is the key. So blue are control, green is non-AD, and red is AD pathology. What we do see here, it doesn't look good, all right? We suggest that for clinical diagnosis that's given from 8 years to 2 years before someone dies, there's not a good correlation between the diagnosis and the blood levels of this marker. So what could be the problem? So we went forward to say, okay, what about we don't use the diagnosis that was given by clinicians during life, but use the diagnosis that was given after death, right, at postmortem. That is the surest way to say someone has a disease. And then when you recolor code them based on the diagnosis given after death, then you get a better view, a very, very accurate view, which suggests that the marker associates much better with the key biological changes that are seen after death than the syndromic ways of diagnosis that are done in the clinic. But then even here, you can see that some individuals have a higher level. I mean, they're non-AD, statistically difference, but why do we have some people sticking out here with increasing levels that do overlap with those in AD? So we looked at these ones. And what we do see that we look at the pathological reports, some individuals had secondary diagnosis. So they have the primary diagnosis is A, but then they can have another diagnosis who comes in as B. So here, those who were like pure AD, that no secondary diagnosis was given. But these ones were given secondary diagnosis, those in other words. So they have AD plus some other disease. Those ones have the same levels as you have for AD. So it doesn't matter the co-pathology that you do have. It doesn't matter if you have any other disease. It's going to tell you that you have AD. That's what this test does. And that those who pathologically did not have any other form of AD, like any evidence of AD, but other diseases, you have way lower. And they match with the controls, which is a pretty good thing. So 8 years before someone dies, there was a 97.4% chance that this marker would tell you that the individual has AD or not, which is pretty accurate. But then we also looked at longitudinal changes. So what happens that if someone has the measure, if you take blood from someone, you measure it today, what happens a year later? What happens 2 years later? What happens in the future? And we got the opportunity to use the ADNI. So there's a really well-characterized cohort from the US and Canada. So what we did was that if we, based on cutoffs, we say, okay, someone has high levels, someone has low levels of plasma phosphatile. Those are the blue. So from the baseline, the blue were all controls, right? They were all negative. So we say they don't have anything. But then over time, you get to see that some who had a little bit higher levels of phosphatile at baseline, although they were controls, if you monitor them for over 5 years, they're starting to decline. So it means they're starting to move on to dementia. And then those who had cognitive decline, so MCI, you get to see that a lot more of those were positive for the marker at baseline from the start. 5 years later, continue to decline really sharp towards dementia, which suggests that the initial measure, so whatever measure that you're given today, if your blood were to be analyzed for this marker, could tell you quite scarily what's going to happen in the future. And if you look at longitudinal changes, the changes per year are really tiny, which tells you that the assay is really stable and that the changes that do occur are also biologically meaningful. We have like about 0.2 or 0.3 picogram per ml changes every year that across groups for each individual. And then if you model these changes based on looking at the life course of this marker, if you take individuals from controls to MCI to disease, what happens longitudinally? You can see that the changes that are occurring with the blood is practically the same change that's occurring with the CSF, and that they also follow with the amyloid beta changes, so that they meaningfully tell you something about what's happening at the biological point. More recently, there have been new markers that have been discussed, that they've been phosphatile 217, and we have been working on that. So one of the things we did, starting from, this was a study in CSF, we came up with a blood studies that are coming out soon, that instead of just taking, let's say, a 217 marker and comparing to what are currently being used, in the clinic for 1A1. What we did, we took a different approach because we believe that, as I told you initially, the more N-terminal you go, the better, the more earlier, the more abnormal the marker becomes earlier in the disease course. So we compared two new markers, one 217 marker that's looking at the N-terminal part, the left part, as I told you initially, and another one, which was measuring 1A1. But what we did was that we used the same immunoassay, so the same partner antibodies at the N-terminal level, and then we compare them to the commercially available ones that have been, cleared by the FDA and have been used in clinical trials now, which target the mid region of the protein. And what we do see is this: that the two new ones, the N-terminal directed ones, they better predict amyloid abnormalities. They better predict changes that you can see between MCI, for example. So if someone comes to the clinic with cognitive complaints, you're not sure if they have Alzheimer's or they have other form of dementia. It tells you that these new markers can, to equal measures, tell you what you see, that they're both accurate, that we don't see significant difference between them. However, they both become abnormal earlier in the disease course, ahead of what we currently use as gold standards in the clinic. So it means that these markers can tell you before someone is closer to dementia what is going to happen. And however, when the person reaches dementia, all the three markers are equally accurate, as we see here, that between Alzheimer's dementia and other forms of dementia, they are all actually about 100% accurate to differentiate between them. And another marker, the phosphorylated 231. So this is quite a really beautiful cohort that we studied from Barcelona that we recently published in Embo Molecular Medicine, that if you model the changes, so these are different CSF markers. So I just told you about 217, two different 1A1s, right? The one that targets the middle and one that targets the N-terminus. And that's we now add 231 to it. We have total tau also there, that you model the changes in preclinical. So preclinical are individuals whom, when you measure the CSF or if you do brain imaging, they have biological evidence of Alzheimer's dementia. However, clinically, they have no evidence. Their cognition is intact. So if you have a grandma at home, they're not showing any sign of being forgetful or anything. However, biologically, they might be showing changes ahead of what happens. That if you model these changes, what we see is that 231 starts to rise pretty early. They all start to rise quite nicely. But all these new markers, which are targeting the N-terms, except the 231, but now we have the N-terminal version also, they rise pretty early and they keep going really fast. But they rise ahead of the current gold standards one that we have, the 1A1, the one in green, both the mid-region and both the total tau. They do not rise much early. So it means that very early in the disease, these are not the best markers, that the new ones that we're showing are really much better. And even in blood, these new marker, the 1A1, which at that time, that's the only one that we had, that also increases ahead of what you see with the CSF current gold standard assay. So what it means is that the new markers we have identified are able to tell you Alzheimer's dementia way earlier than the industry standard measures that we currently use. And the advantages are that they will be able to tell you what the current one cannot tell you. Someone who is likely going to develop the disease in the future, and that you could start treatment earlier, and that you could use these individuals to test new drugs, because if you target the disease early, likely they'll be able to address them. And 231, we just looked at those in blood, and some quite scary findings that if you look at the amyloid positivity and you stage these amyloid changes, that they appear to come ahead, way ahead of 1A1, even 217 in CSF. And if you look at in primary care, 1A1 and 231, so this primary care, individuals who go to the GP or who come to the primary care physician, they have no evidence and no biological test is done on them. You don't have any psychiatrist or neurologist or any clinician who is a specialist to look at them. This is based on just what the primary care physician is giving. What we see here is that the test can give you an idea who should be moving on to be referred to see a neurologist or a psychiatrist, or who should not. Because sometimes cognitive impairment could be due to anything. It could be due to dementia, and it could also be due to some transient changes. So with all these things that we've talked about, how could these be used in a clinic? What is a clinical context of use? What we think could be happening is this. Individuals don't go to the clinic unless they have cognitive impairment, right? So those who come to the clinic with cognitive impairment, this is our proposal, that what we have shown is that if you have high phosphatide, it means you have higher amyloid, that they could be first assessed for phosphatide, any of the ones, the three that we've talked about. Currently, there's a Karikari, so a commercial company. We collaborated with them to build the 101 kit into a commercial one, so it's not commercially available. If someone has a measure above the cutoff value, then they're likely to have Alzheimer's. So the clinician can start looking at them for Alzheimer's. If they do not have Alzheimer's disease, then, or they don't have low levels of phosphatide, then they don't have Alzheimer's. Then you can measure plasma neurofilament light. Neurofilament is a good measure of neurodegeneration. If it is yes, it means that they have a degenerative disease, but that is not due to Alzheimer's, because if they had Alzheimer's, they would have high level of phosphatide. If neither of these is positive, then they are not likely to have neurodegeneration. So you could start looking at them for non-neurodegenerative impairments that are causing the cognitive decline. And that could be quite a nice algorithm to help clinicians diagnose. And in clinical trials, what could happen is that currently is really expensive, extremely expensive, because they use PET, and PET scans are really expensive. They could cost about $5,000 per single scan, and you have to do several ones, of course. So if we look at, okay, is plasma phosphatide, 101 or any other ones, increased? If it's yes, then individuals are likely to have Alzheimer's disease. And because now we have these markers that can identify very early changes in the disease, then it means that we can be sure we can include individuals in the preclinical stage, those who do not show any clinical symptoms. However, biologically, they have evidence. Then those will be the best ones to target, because the disease is not pronounced. So if you have any drug, it's likely to be more able to target these deposits of tau or amyloid. So if they have Alzheimer's, then you can include them. One option would be to confirm with CSF or PET, right? You say, okay, you identify several individuals, you screen them, you identify those with high increase of phosphatide, and then you confirm that with PET or CSF, which means that instead of, let's say, screening 5,000 people with PET, let's say each at $5,000 per single scan, you will be able to now screen 5,000 with phosphatide, and that, which is much, much more cheaper and quicker, and that you select only those with positive phosphatide to confirm with PET or CSF, and you enroll these individuals. And then you could also use the same marker as an outcome measure, because if the individuals had high levels of phosphatide, which meant that they had high disease, or they were advancing a disease, what it also means is that if your drug is able to reverse the disease phenotype, then the marker should also now starting to decline. So you could use that as a way to test if drugs have been useful. One thing that I always like to talk about, because I come from Africa, this is one of the things I feel is that whatever I do, whatever I contribute to the body of science, if my people do not find a benefit for it, then I have failed. So what I always think about, how could these markers be useful in developing country settings? One, they could be used in epidemiological studies, in population-based studies also, and also in clinical investigations. Currently, we have no evidence of the statistics of dementia in Africa. We don't know, except maybe South Africa and a few developed ones. But we don't have any key evidence. We could use these markers to screen. We don't even know that if the basis of Alzheimer's, as we know in developed countries, is the same, because there are some social-cultural aspects, there are some lifestyle differences, there are some genetic differences. We don't know much about this. So if we use these markers, maybe we can start looking at the biological determination. The advantages are that the cost is low. It increases the speed of investigation because it's blood. You just take the blood and then you process, you just measure quickly. It's simple, and it provides biological evidence of the disease, and it could help with differential diagnosis. But one question still remains, how feasible could this be done? There's a nice study here that a colleague of mine that they've done recently, where they used um, um, either normal filter paper, right? So dry blood spot. You just take blood, dip it on filter paper. It dries. You cut the shape of blood. You just keep it. You can store it forever. When you're ready to use, you just dip it in buffer, and then the blood just goes back into the buffer. You centrifuge it and you use, which means that this could be useful for field studies. In this other one, which is like a Noviplex card, so it's a card that can separate the different components of blood. But doing that within the card, you don't need a centrifuge, because some field studies or some remote hospitals don't have centrifuge or cooling, equipment, no refrigeration that can be done. And that if you also, this is the regular that you take blood, you let it sit for 2 hours, and then you centrifuge. You compare these ones. Basically, what we're showing is that if you compare all of these, if you measure, if that you do it as we do it normally, you take blood, 2 hours you process, and you compare with using any of these. So this centrifugation is if you delay for 48 hours. So assuming you take the measure from a remote site, and it takes you 48 hours to come to the lab, then you can actually do that. You can just use any of these methods compared to the regular process that is globally accepted. The measures that you get from them, they correlate really perfectly with the standard measures, which tells you that in settings where it's more difficult to process blood, collect them and centrifuge and refrigerate or freeze, you could use any of these methods and they will be as good as the regular approaches. Okay, so in conclusion, just talked about that way markers for phosphatile in blood, and they correlate with all the biological evidence that we have for Alzheimer's, both in sporadic and also in genetic basis of disease. And they appear to be quite earlier than the classical markers that we currently have. These findings have been also validated by some other colleagues who have used different tests that have been developed independently, and that we've been in collaboration with a company, a biotech company that's developed one of the markers into a commercial kit. So it means that now they're accessible, anyone can buy them and use, and that these markers could be useful for clinical trials and also in regular routine memory clinics, in also primary care as well, and that their efforts are being made to enable these markers to be used in developing countries. So I am grateful to all the funders, these and several other ones. Bright Focus Foundation, that's giving me a research fellowship currently, the Alzheimer Foundation in Sweden, the Yawn Foundation, which is the Heart Foundation, the Dementia Foundation in Sweden, and several other ones who have been kind enough to fund me and all the work that we've been doing here. And I thank all the colleagues of mine who have been extremely helpful with the work that we've been doing here. And thank you for listening to.
Fantastic. Thank you very much, Thomas, for a wonderful presentation. It's so great. I mean, I've looked at the papers published, but now it's much clearer to me. I used to have a lot of questions then, but now it's clear, and I'm more convinced now. So we are now open for questions. If you do have any question, I think this is the time to ask or send the question through the Q&A button. But I'll start with asking, I mean, this whole thing is, within 1 year, it's more like the Cambrian explosion. Tell me, how have you managed to get all these different things, done? I mean, because this presentation is recorded, and there are a lot of early career researchers, people, wanting to join science, and, it would be nice to hear your story.
Well, there isn't much to say. I don't know how it all happened, but I feel it's, if there's anything I could say, is about just read between the lines. In the sciences, there are hard questions that people have to ask. There are things that people get to think like it's a done deal, that is too difficult to do. It doesn't hurt to try. Oftentimes you would fail, but also even in the failures of your experiments that not working what you do, what you want to find, there will be some evidence in there that if you follow. Because what I think is that when we often get so engrossed in having a hypothesis, and we want to make sure that whatever we find fits the hypothesis. So we are so narrow-minded. I want to see this, that's that. But if you knew it, you wouldn't be testing. So in the course of testing, and although you're convinced that you want to see X, just be open to all the other alphabets, that there might be another one that you might see. It wouldn't be the X, because you don't truly know what it is. That is why you call it science, and that's why you're researching, and you might find something else.
Absolutely, absolutely. I was thinking from the angle of the science bit of it, with all these new issues coming up with football, CTE, kind of serving as risk factor and everything. Have you guys started thinking about seeing if any of your kind of markers could predict that?
Yep.
Is there anything ongoing?
Yes. So we've done one cohort with CTE, because we do everything blinded. Like when we collaborate with people, we don't want to see, we just want codes, so blinded codes. So they're looking at the analysis of CTE. I think there might be some evidence of increased phosphatile, and it could be that these increase could be with some specific markers and not all of them.
Mm-hmm.
So yes, that we can see these markers in CTE. However, one thing that I'm also keen to see is that, are they showing CTE or they're showing AD in CTE? Because one of the things we've seen is that when you have any efforts that lead to the blood-brain barrier being pressured, you can see a gush in phosphatile coming into the blood. But this clears out really quickly over 24 hours. So, for example, when some people are given anesthesia during heart surgery, for example, or people who are undergoing stroke, at the point of stroke, you have a super fast increase in phosphatile and total tau also coming into the blood at the time of it. However, by 24 hours, it's normalized. The other markers that are currently being used, like NFL, neurofilament, they don't come, they don't come into blood until about 3 days, like they peak about 3 days, by which time phosphatile has come in and gone. So you could use this as markers. But however, they also tell you that you have to be cautious if you're just using phosphatile as a one-time measure to say someone has AD, because it's likely that, if it's less likely, if not many people are going to have any impact to their brains. But if this has happened at the time of that event or the day before or something, you have to be cautious that it could be due to a biological effect, of what's happening in the brain due to something non-AD related.
Fantastic, fantastic. So we've got a question from someone. He asks, Dr. Thomas, how do you get to do a lot within a very short time?
Because it's because I've been quite blessed to have a lot of people with me. We're a very enriched team that we split the work among ourselves. We know what we're doing, so we can ask everyone to do different things at one point in time. I am more interested in the development, in the new markers, in the biological aspect. What happens? What about new marker? What does it do? However, we have technicians and other who help to also, to, once we have identified these changes and it's been cleared, to also help us run these analyses in the thousands and thousands of patient samples that we do get to confirm that what we think we're seeing is true. And another advantage is that we are not so much constrained financially. I have worked in Ghana, I have worked elsewhere, that every time you have to, the first thing you think about is the money. We are not the richest, of course, but then if you don't have to think of the money all the time, there's a lot you could do, because you just focus on the science and what and if and how and why. Those are the things that push you, that the financial constraints get pushed to the background.
I wish this is everywhere. I mean, if only, money is not an issue everywhere, then science would just be so, so great.
Yeah.
Yeah. So I was thinking, so by the way, people, you can send questions. We still have some few minutes. So I was thinking in terms of I've always seen tau as like a protein that responds to cellular distress. And here, in this work, you're showing so specific that with AD, you've got an increase in this marker. So now I was kind of thinking, do you therefore think in terms of these other tauopathies, there would also be some specific markers? And if that's the case, perhaps we have to stop thinking about having a therapy that can be one size fitted all, if you see
Yep.
what I mean.
Yes, that's very true. Because now we have over 80 phosphorylatable sites in tau, but then we haven't studied all of them. Some seem to be phosphorylated as a consequence of others being so they are clusters of phosphorylatable sites. And so for the non-AD tauopathies, they are quite difficult to deal with. That almost all the markers we've tested and we've looked at from CSF to blood, they all get to be specific for AD. And in terms of truncation, which is a bit more difficult to track, potentially there might be some. But one thing we've seen that I'm not showing that data here is that if you look at postmortem brains, you don't see a lot of truncation in the non-AD tauopathies. The truncations are a lot more in AD. So my hypothesis is that in the brain, you could have a lot of processing of tau occurs in AD, and that might be the reason why you see some of these fragments being released, coming to the CSF and also into blood. And maybe for the other diseases, the processing is a lot less. So although we're seeing phosphorylation of tau in the brain in all these diseases, how they end up to become markers, pathophysiologically in biofluids is a bit more difficult to find. It's what we are focusing on. We feel that now AD seems quite well sorted. Maybe we should be focusing on those ones now, also to understand why that such differences are difficult to find.
Absolutely, absolutely. I mean, there is definitely a lot to do here, which is quite exciting, I guess, for you and for the field as well. We've got a question. The person said the blood sample taken from the tip of the finger, is it reliable? Is it enough?
Well, reliable is number one, enough. So enough in terms of the amount, right?
Mm-hmm.
Well, I think you would need a lot more. I mean, not just like, just tip drop like that. Maybe you need quite a bit more. But then from the data that we've shown, this is showed that it means that if you just take the blood and then you let it dry, get like a dry blood spot and keep it, you can process it forever. And I think this would be a huge thing for settings where centrifugation, freezing, electrical power outages is a big deal, that you could basically just have such a way to take samples, maybe take multiple times, instead of taking like a single drip, just do multiple times, just you have a lot. And that, or you can just do a blood draw, have a certain volume and just dip it onto blood spot, make it into spots and let these dry. Reliability from this marker, at least for neurofilament light, we're showing that it's reliable. The question is, are they reliable for the other markers that we know for blood, for tau and for phospho-tau, for example? And that's one thing we have to do. But at least it's a good thing, where a good point to start to show that potentially, if we do know all the challenges that people do have that we'll know of in different settings, in multiple settings, there would not be an impediment to being able to partake in such sort of studies.
Absolutely. I mean, one of the things that I was thinking while you're talking is with sporadic AD, where, especially not the one with ApoE, with double allele for ApoE4, I would imagine that this kind of markers would help in if in the future, perhaps, people are being studied and, there's an increase in this there, then physicians could advise them in terms of lifestyle changes or something that perhaps could help in either slowing the progression or
Yeah.
something else. So this is really, really great. Um, yeah. So there's a question again from one of the audience. It says, is there possibilities that after spinal cord injury there will be increase in p-tau?
I would say yes, as a possibility, that and I would imagine also depends on what kind of spinal cord injury that's happened. That because we have the CSF, it's in the spinal cord is like is in close contact with the brain in terms of like CSF from blood to brain. That if we have an effect coming from the brain, it goes through the spinal drain, and that if the injury is affected in the spinal cord locally without any effect to any brain related effects, then you might see a change that comes to blood, but that would not be related to Alzheimer's in the brain.
Absolutely.
So that would be like a false positive.
Mm-hmm.
Oh, so that would be a really cool thing to look at. That what if, because we have, you have this sort of chain, brain, CSF, blood. What happens if something occurs midway and not at the source? Of course, that would not mean it's AD, but from a biological perspective, what would that mean?
Absolutely, absolutely. I mean, we've run out of time, but one of the things I was even thinking is in terms of these fragments that you pick up in the blood, it kind of tells you that somehow the tau escapes from the brain, from the CSF to the blood, and it's kind of, I mean, for those interested in how these things get out of the brain, I guess it's something to look out for. How does that happen? You know, the mechanism, etc.
Yeah, it's one of the things that I'm quite interested in.
Cool. Thank you very much, Thomas. It's been a great presentation, and I guess it's been great for the field as well to find these new biomarkers, which hopefully will speed up discovery for either, diagnosis or for therapy, etc. And as someone who is also who came from Africa, I guess you'll get another invitation in the future to talk about how you're doing it and how other African scientists could do this.
Always available and do whatever I can to help. And also, just as we all know how it happens, ready to help anyone who reaches out how we can do it, and any advice, any supports, we can help.
Fantastic. All right. Thank you very much and take care.
Bye-bye.
Bye-bye.